Today, we'll see the another unit, that is unit number 3, chromosome structures and variations. Under chromosome structures and variations, there is another subpoint, that is the alteration in a genetic makeup and its significance. Under that one, we'll see the change in chromosome number, that is the euploidy and aneuploidy. First of all, we'll see the introduction. The chromosomes of each species has a characteristic morphology, that means the structure and number, but sometimes due to certain accident or irregularities at the time of cell division or crossing over or fertilization, some alterations in the morphology and number of chromosomes takes place. There are two types of variations, that is the first of all, we will see the ploidy. Under ploidy, there are two subpoints, that is the euploidy and aneuploidy. Under euploidy, there are further three parts: monoploidy, diploidy, and polyploidy. Under polyploidy, further it is divided into autopolyploidy and allopolyploidy. While under the aneuploidy, further it is divided into two parts, that is the hypoploidy and hyperploidy. Under hypoploidy, further it is divided into monosomy and nulsomy, while the hyperploidy it is divided into a trisomy and tetrasomy. Variation in chromosome numbers. The genetic number constituent a basic set of chromosome called as a genome. A gamete cell contains a single genome and it is called as haploid. When haploid gametes of both sexes unite in the process of fertilization, a diploid zygote with a two genome is formed. The diploid zygote undergoes the embryological development and forms an adult animal, which upon attaining the sexual maturity produces the haploid gametes. Sometimes the size and the number of chromosomes in the haploid or diploid cells does not remains constant same but alter variously variation number of sets of chromosome. It is encountered in a nature. First of all, we will see the euploidy. The term euploidy, that is the Greek even or a true, ploidy means unit. It designates the genome containing the whole set of chromosomes. The euploids are these organisms which contains a balanced set or set of chromosome in any number. The euploid may be of following types, that is the monoploids, diploids, triploids. We'll see one by one. The first of all, we will see the monoploids. The monoploids organisms have a one set of chromosome or a genome, that is the N, in the nuclei of their body cells. For example, terran insects. While in a diploid, the diploid organisms having a two sets of chromosomes, that is the twice N, and therefore two copies of the genes, especially in a somatic cells. While in a triploids, the triploid organisms have three sets of chromosomes or a genome, that is a thrice N, in the nuclei of their body cells. It is the union of monoploid gamete with diploid gamete. Due to extra set of chromosome, genetically unbalanced gametes formed. The triploids are usually sterile. For example, some cells of bird, reptiles, mans, and other vertebra. Then we will see the tetraploids. The organism with four genomes, that means the 4N in the nuclei of their somatic cells, it is called as tetraploids. The tetraploidy arises by somatic doubling of chromosome number. It can be induced by exposure to chemicals such as xanthonaphtine and veratine, and also exposure to heat and cold. For example, wheat, chrysanthemum, and solanum. Then we will see the polyploidy. Any organism with more than two genomes, that means the twice N, it is called as polyploidy. The flowers are polyploids. For example, in a wheat, the hexaploid, that means 6N. In a strawberries, octaploid, that means 8n. The polyploids may be of following two types. Autopolyploids. The autopolyploids are those polyploids which consist of some basic sets of chromosomes multiplied. For example, if a diploid species has two similar sets of chromosomes or genomes, that is capital A, and autotriploid will have three similar genomes, that is the capital A, and autotetraploid will have four such genomes, that is the capital A. Since an autotriploid remains sterile and cannot produce seed, therefore it has a great commercial value in producing the seedless varieties of economic plants. For example, in Japan, the Chiara produced seedless watermelons, that is the autotriploids. The autopolyploidy have been induced in many plants and animal cells by artificial means such as a chemical, for example, radium and X-rays and temperature shocks can be used for development of autopolyploids. Then we will see the allopolyploidy. When the polyploidy results due to doubling of chromosome number in a F1 hybrids which derived from two distinctly different species, it is called as allopolyploids. For example, the species X crossed with species Y. The species X is the diploid while the species Y is also diploid. There is a formation of F1 AB, that is the diploid strata hybrid. While you treat the colchicine, then it becomes a fertile, that is the ampidiploid tetraploid. Then we will see the aneuploidy. The variation in chromosome number many occurs which do not involve the whole set of chromosomes, only the part of set. The term aneuploid, that is the uneven, and ploid means unit, is given to variation of this nature, and the suffix somic is a part of their nomenclature. The aneuploid may be of following types, that is the monosomic, nullsomic, and so on. First of all, we will see the monosomic, that is the diploid organism which are missing are the chromosome of single pair are monosomic with the genomic formula is twice n minus 1. A monosomic individual forms a gamete of two types, n and n minus 1. The n minus 1 gamete do not survive in plants, but in animals they may cause the genetic imbalance. Then we will see the nullsomics. An organisms which has lost the chromosome pair is a nullsomic. The nullsomic organism has the genomic formula, that is the twice n minus 2. A nullsomic diploid often does not survive. However, in a nullsomic polyploidy, for example, hexaploids with 6x minus 2 may survive but exhibits reduced vigor and fertility. Then we will see the trisomics. The diploids which have one extra chromosomes are represented by a chromosomal formula, that is the twice n plus 1. One of the pair of chromosome has an extra number so that a trivalent structure formed a trisomy can produce the different phenotypes depending upon which the chromosome of complement is present in a triplicate. Then we will see the double trisomics. In a diploid organisms, when the two different chromosomes are represented in a triplicate, the double trisomy cause a great genetic imbalance and has a genomic formula is twice n plus 1 plus 1. While in a tetrasomics, the diploid organisms having two extra chromosomes are known as tetrasomics. They have a genomic formula, that is the twice n plus 2.